The events of muscle contraction are summarized in what is known as the sliding filament theory. We have already reviewed the initial steps, which begin with the arrival of the nerve signal and the release of the neurotransmitter acetylcholine. The acetylcholine binds to the plasma membrane protein receptors, resulting in the opening of ion channels within the motor end plate. Sodium rushes into the cell and potassium leaves, resulting in depolarization of the membrane at the motor end plate. When the charge on the intracellular side of the end plate reaches a threshold level, ion channels open across the sarcolemma, leading to a total membrane depolarization known as action potential. The action potential is propagated down the T-tubules. As you recall, the T-tubules are invaginations of the sarcolemma that penetrate into the interior of the cell and always lie between paired terminal cisternae of the sarcoplasmic reticulum. The change in electrical potential of the T-tubules causes the release of ionic calcium from the terminal cisternae. Ionic calcium binds to the protein troponin. The result of this binding is a reconfiguring of the shape of tropomyosin, resulting in the exposure of the active sites on actin that bind with myosin. The resting or activation position of myosin occurs as a result of the breakdown of ATP. The ATP binding site on the cross bridges contain ATPase, an enzyme that facilitates the breaking of a phosphate bond of ATP, leading to the subsequent release of energy. This process is probably more clear as we look at the next step in the sliding filament theory. With the exposed active sites on the actin, the cross bridges attach to the actin and perform a power stroke that pulls and slides the thin filament over the thick filament, shortening the sarcomere. This is muscle contraction. Following the power stroke, ADP is released from the cross bridge and replaced with a molecule of ATP that is immediately hydrolyzed, returning the cross bridge to an activated position, ready to once again attach to actin and perform another power stroke. So it should be noted that ATP is required to position the myosin molecule for the power stroke. This process is called cocking and activation of the myosin head, as described in what we have labeled Step 10. This animation shows the physiological events associated with the actin-myosin interaction of the sliding filament theory. Use the legend in the lower right corner to try to follow the sequence of events. So there needs to be a mechanism that stops muscle contraction and returns the muscle to its resting state. This starts with the nerve no longer releasing neurotransmitter. In the synaptic cleft is the enzyme acetylcholinesterase. This enzyme breaks down acetylcholine to its component parts that are taken up and recycled in the neuron. Without the presence of acetylcholine in the synaptic cleft, the sodium gates close. Also, without the influx of sodium across the membrane, calcium is reabsorbed by the sarcoplasmic reticulum without being released by the terminal cisternae. Since the calcium ultimately results in the changing of the tropomyosin configuration, the lack of calcium ultimately blocks the myosin-actin interaction, stopping muscle contraction. So now muscle contraction has stopped, but the cell is still depolarized. The cell must now be repolarized. Repolarization is first established by opening and then slowly closing potassium gates along the sarcolemma. As you may recall, potassium ions are positively charged. Potassium ions are also in greater concentration inside of the cell versus outside of the cell. Therefore, when these gates open, potassium diffuses out of the cell, taking their positive charges with them. This leads to the inside of the cell becoming more negative again. Ultimately, the cell becomes electrically polarized. Although the cell is now electrically polarized, ionic balance has not been restored. The cell still has too much sodium within it and has lost a significant amount of potassium. The ATP-driven sodium-potassium pumps, mediated by specialized transmembrane proteins, work to expel sodium and recapture potassium at a 3:2 ratio, ultimately restoring the ionic concentrations. The muscle is now at rest and fully ready to receive another signal from the nerve.